Morning everyone. This is GS Academy Rampuram Chennai. We are giving online and offline courses. Helpline is given here. CSIR Net. This is Life Science Unit 4 Part 3. This class is about basics of cell communication. Let us see the first one. Autocrine signaling. What is autocrine signaling in the sense? See the cell. This is a cell. This is releasing extracellular mediator that binds on the receptor site of the same cell. So it is called as auto signaling or self signaling. And if you see where it takes place in the sense, let us take this is a liver. If there is any damage occur, that will be rectified by this auto signaling. How in the sense, the cells which is nearer to this divides into two and heal this part. So why this is called as autocrine signaling or self signaling. This is not only in liver, and also if any bacteria enters inside the body of any organism, or if it is a human body in the sense, the T lymphocytes will be produced in more amount to kill the bacteria. And this also takes place in macrophages. The production of macrophages will be more by auto signaling. See, for example, this macrophage give a signal to this cell itself to divide. So this macrophage divide into two. Again, these two are giving signal to them. So it is into four. Like that, this will multiply to kill the organisms, to kill the disease causing organisms. This is also happens in the lymphocytes and in the platelets. The platelets proliferate and production of collagen takes place when there is any wound occur or any blood loss. At that time, this will produce in large amount to check or to stop the blood flow. And another one important thing what you are going to see is the cancer cells. How the cancer cells are proliferating in the sense, the cancer cell divides through auto signaling. So first two cells, then these two produce two more, then these two more produce another two more. So like this the proliferation of cell goes on, and the cancer cells are forming a numb-like structure, okay, and at last it kills the organism. Next one, let us see what is paracrine signaling. This is very interesting signaling. How in the sense, see, this is a cell what you are seeing. This is a secretory cell that is secreting the mediator, and these mediators are received by the adjacent target cell, and then the work will be done. Okay, what message is passed, that will be done. You can see here the nitric oxide is a significant paracrine signaling molecule. This nitrous oxide can diffuse directly across the plasma membrane to its target cells. See, if it is releasing the nitric oxide in the sense, this directly diffuses through the plasma membrane and it enter into the target cells. Okay, for example, steroid hormones are released in the sense that will be directly enter into the adjacent cell through this plasma membrane. And another one is carbon monoxide. This carbon monoxide also serves as signaling molecules in the nervous system. Okay, so this also very, very important. So make a note. Carbon monoxide gas, okay, also serves as signaling in the nervous system. And another one you can see, icosanoids. It is prostaglandins, prostacyclin, thromboxanes, and leukotrienes. These are a class of lipids that function in paracrine and autocrine signaling. These are take part in the paracrine as well as the autocrine signaling. Okay, you can see here what is paracrine and this is autocrine. What you are seeing here is a cell that is secreting that mediators are received by the receptors of the same cell, and the work is done there. Okay, that's what we have seen in the liver cell. Next is, what is justagrain signaling? What is justagrain signaling in the sense? This communication junction links the intracellular compartments of two adjacent cells. What you can see in the sense is, for example, plant cell, if we take the plant cell, see there is a gap which is present between the two adjacent cells. There is a connection between the two adjacent cells that is called as plasmodesmata. In the previous class we have seen. Okay, so it's a passage between the two adjacent cells that is called as plasmodesmata. So through this plasmodesmata, these mediators are passed from one cell to another cell. So this is called as justagrain signaling. Justagrain signaling. As well as you can see this one in the nerve synapse. See, this is a cyton and this is axon. This is nerve terminal. And here another nerve in the sense. This part is called as synapse. So here the materials will be transmitted from one neuron to another neuron. Okay, so this is the example of justagrain signaling. So if it is comes in the examination in the sense, that is electrical synapses or electrically conductive gap function between the neurons. Okay, and here also in plants it is plasmodesmata, the gap junction in animals, as well as in plants if you see it is plasmodesmata. Okay. Next one, if you see it is what is endocrine signaling? Endocrine signaling in the sense, see if you take this is the glands in the sense, glandular cell. If you take this is a glandular cell, for example, adrenal gland, if you take, the adrenal gland is releasing the hormone. That hormone is carried by the blood vessel and it is giving to the target cell. The target cell is receiving this one through the receptor and it is doing its function. Okay, so this is called as endocrine signaling. What is paracrine signaling? The adjacent cell, the secretion will be secreted out, that will be received by the adjacent cell. And autocrine in the sense, the autocrine, the cell is releasing the mediators that will be received by the same cell. Okay, so we have seen this one, and this is the first one we have seen, and this is the second one, and the fourth one is justagrain, where it is happens I have shown you. And the next one is, fifth one is quorum sensing. What is quorum sensing in the sense? See, if a bacteria enter inside the body, that proliferates, okay, and it is it covers with the biofilm formation, and this bacteria is very, very virulent inside this cell, and inside the cell it multiplies and it causes the diseases. Okay, and the victim will be suffer. Whereas this will be, if it is more, this will be sensed by the body and it is producing the quorum sensing inhibitors. What this quorum sensing inhibitor does in the sense, it kills the bacteria, or it inhibit the proliferation of the bacteria. Okay, so the person will be safe from the disease. Or else if the person is taking the medicine that will inhibit the growth of the bacteria, so the person will be safe. So this is called as quorum sensing. So what is happening here in the quorum sensing in the sense, they enable this bacteria will be restrict to the expression of the certain genes to the high cell densities at which the resulting the phenotype will be most beneficial. And many species of the bacteria use the sensing to coordinate and gene expression according to the density of the total population. So this will be suppressed or inhibited by the by the quorum sensing inhibitors released by the body, or from the medicine what the person is taking, that will be inhibited. The proliferation of the bacteria will be inhibited. Okay. One more important point is gram-negative bacteria produces N-acyl homoserine lactones. AHL means acyl homoserine lactones as their signaling molecule. So if the question comes, what is the signaling molecule for N-acyl, for gram-negative bacteria, in the sense you have to answer N-acyl homoserine lactones. Okay. And you can see one more important point, that is bioluminescent bacterium. Bioluminescent bacterium, it is Alivibro fishery. This is called as Alivibro fishery. Alivibrio fishery. What this bacteria, in the sense, this is a bioluminescent bacteria. When it is in less concentration, we cannot see the bioluminescence, and if it is in more concentration, that is about 1011 cells per ml, in the sense, we can see the bioluminescence. Usually where is this bacteria live, in the sense, that is Alivibrio fishery, where it lives, in the sense, it lives in the body of the squid. Okay, it is a common salism. More than common salism, we can see this is a mutualistic symbiont in the photo 4 of the squid. That squid is called as Hawaiian bobtail squid. Okay, so what is this bacteria as well as this squid is doing, in the sense, mutualistic symbiosis. Okay, the relationship between this bacteria and the squid. And when this fishery, that is Alivibrio fishery, are in free living, in the sense, auto-inducer, which is in lower concentration, and I told you if it is luminescent only in the higher concentration. What is that concentration, in the sense? About 1011 cells per ml. Okay, and if you see this one, Alivibrio fishery bioluminescence is regulated by AHLs. I told you already what is AHL. AHL, in the sense, N-acyl homoserine lactones. Okay, N-acyl homoserine lactones. Okay, which is a product of LUX-L gene, whose transcription is regulated by LUX-R activator. Okay, so what is this AHL, in the sense? AHL is the product of LUX-L gene. Okay, product of LUX-L gene, whose transcription of this is regulated by LUX-R. Okay, LUX-R. What you are seeing here is the Vibrio fishery, bioluminescent bacteria. It is symbiotic with bobtail squid. This is the electron microscope picture of this Vibrio fishery. Okay, so let us recall once again, and what we have seen is what is autocrine? Autocrine, in the sense, the cell secretes the mediator that will be acted on or received on the same cell through the receptor, and the work will be done. One example what we have seen is the liver cell. Okay, liver cell, if there is any damage takes place, that will be rectified by the same cell of the liver through the autocrine signaling, or it is called as self signaling. And what we have seen in the next one, it is also takes place in the T lymphocytes, macrophages, and platelets. Okay, so all these cells will be at the need. If it is need, then only they produce, otherwise there won't be any division of the particular cells. Okay, so the auto signaling will be stopped when it is not required, and auto signaling will be starts when it is required. The body required only, that will be done. Next one, if you see, Paracrine signaling, that is the secretor cell secrete the mediators that will be received by the receptors of the adjacent target cell, that the work will be done what it is signaled. Okay, and for this I have shown you all those things. And the next one, if you see the justacrine takes place between the two cells. You can see here, which is through the ligand. Through the ligand, the signal will be given. Okay, that is in plants. It is through plasmodesmata, and in between the neuron, it takes place. The gap in between the gap of the neuron, it takes place. And fourth one, if you see, that is endocrine signaling, in the sense the endocrine glands, they produce or secrete the secretion, that is the hormone, which is taken up by the blood, and that reaches the target cell. Fifth one is quorum sensing, that is by the bacteria, and the gram negative bacteria produces N-acyl homoserine lactones as their signaling molecule. Okay, this is very important, so make a note. And this bacteria, that is bioluminescent bacteria, Vibrio fischeri. This bacteria is a bioluminescent bacteria. This is living in the body of the squid. The squid is called as Hawaiian poptail squid. Okay, and they are living a mutualistic symbiotic relationship. And here another one important thing is the bioluminescence is regulated by AHL, okay, which is a byproduct of the LUX gene, and who is the transcription is regulated by LUX or activator. Okay, so I hope you have understood very nicely all the 5 different signals of cell. Thank you so much for watching this class. Share this video to your friends, those are preparing for CSIR Net, and our address is given here, a helpline also there. You can contact through this, and you can get admission in our. Okay, thank you so much.